Okay, so last time we studied up to here, and I wanted to tell you that these slides are not included, so you can skip these slides. Monoclonal antibodies. Number two, this is also you can skip. No need to prepare this one, and no need to prepare this one. Basically, these four slides I brought in for one of my students. She wanted me to discuss, what are the various researches happening nowadays. About the monoclonal antibodies, about the antibodies used as a medicine. Okay, so that's why I had brought it. It is not in your textbooks. We are not going to discuss all this. So we were discussing cancer research, and that's how I brought these, slides for her in my PPT. So these are not in your book, these four slides. So we'll not prepare them for the midterm or for the final exam. Okay. So we're talking about, so let's start our today's lecture. We're still doing the humoral immunity. We did not finish with the humoral immunity. If you remember last time we spoke about the humoral immunity is basically because of the presence of the antibodies, right? So these antibodies, they circulate in our body fluids, whether that be blood, interstitial fluid, or milk, or blood, or anything, right? Or saliva, tears, right? So these antibodies are the immunity. Because of these antibodies, we call it the humoral immunity. And then, if you remember, I told you that antibodies cannot kill a particular microorganism. You have to remember this forever. Antibodies never kill a microorganism. They simply inactivate the bacteria, inactivate the microorganism, or inactivate the toxins, so that they are further killed by some other cells. That could be your membrane attack complex, or that could be a macrophage. But antibodies cannot kill, because they cannot cross the plasma membrane and enter into the cells. Okay, the way T cells kill the virus infected cells, but your antibodies cannot do all that. Okay, so let's try to know the structure of these antibodies. How do they look like, right? So what are antibodies? So antibodies, as I said, that they're basically proteins by the end of the day. Okay, they're called as the immunoglobulins. Okay, we write them like this: I as in capital, G as a small. Okay, that's how we write the immunoglobulins or the antibodies, which constitute our what? Gamma globulin. If you remember, we spoke about the plasma proteins, right? So these belong to the gamma globulins in our plasma proteins. Okay, they are not formed by the liver. Okay, we spoke about that. They are formed by the plasma cells. Last time we spoke about how a B cell is activated, then it's proliferated, it forms its cone, and then how it forms the plasma cell, and how this plasma cell form a lot of antibodies. We spoke about all that in the previous lecture. So they have a capability to bind to a specific antigen. That is why this part is called as the specific immunity, right? Now let's try to know the structure of the basic structure of an antibody, okay, or basic structure of an immunoglobulin. In a while, we'll talk about different classes of the immunoglobulins, right? These are the different types of the antibodies, or different classes of the antibodies, right? So we'll talk about them in a while, but regardless of any type, you have to remember that this is like a basic structure of any of the antibody, right? So each antibody consists of four polypeptide chains. As you can see here, this is one chain, the green one. This is another chain, the blue one. This is another chain, another blue one. This is one more chain called as the light. So the green ones are called as the lighter chains, and the blue ones are called as the heavier chains, which are nothing but they are the four polypeptide chains which are linked to each other at various places through the disulfide linkage. I want to connect this with the biochemistry. Since you have biochemistry exam also next week coming up, do you remember which type of amino acid is capable of forming the disulfide linkages? Does anyone remember? Have you started protein chapter for upcoming midterm in biochemistry. Which amino acid is there which can form the disulfide linkages, which can help us, in the tertiary structure, or in the the higher structures of the proteins? Does anyone remember that? Aya, Running, did you come across? Don't, you forgot it, right? It's okay, no problem. Cysteine, absolutely, Shata, absolutely, Shata, very good. It's the cysteine, yes, absolutely. In cysteine, you might have studied in proteins that it's the cysteine which has a sulfhydryl group, or a thiol group, right? And this amino acid is special because this amino acids can form a cysteine dimers, as you can see here, disulfide bond, okay, between the polypeptide chains, which can hold the polypeptide chains together. Very important. Thank you, Shatha. Very good. Right? So it's the cysteine amino acid. Okay. So now when we talk about the amino acid, it usually looks like this Y-shaped structure, okay, or sometimes a T-shaped structure. Okay. And now let's look at each polypeptide chain. Each polypeptide chain has a variable region, as you can see here, variable in the light chain, variable in the heavy chain, variable in the heavy chain, variable in the light chain, right? So one side they have this variable region, okay, and on the other side they have this constant region, C for constant, C for constant, C for constant, right? Now what happens, this variable region is important because it is different in different antibodies. So that means IgM will have a different variable region, IgA will have a different variable region, D will have a different variable region. But what is not going to change is this constant region. This constant region is going to be, kind of almost same, okay? But it tells you that to which class of the antibody does a particular immunoglobulin belong to. It defines the function of the antibody, okay, this constant region. And what does this variable region do? Variable region, you might have seen that whenever we want to make an antibody, we want to show that antibody binds to the antigen, we make it something like this. We make it something like this, right? So as you can see here, look at this. So this is the place where antigen can bind to the antibody. So this forms the variable region of an antibody. So this is the variable region of the antibody, right? Which is formed by all the four variable regions of a polypeptide chain. So you can say that variable region is an antigen binding site, okay? And it's made in such a way so that it can fit to a specific antigens. Okay, that is the difference between the variable region and the constant region. We move on to the very important topic, okay, called as, different types of the immunoglobulins. Okay, and what are the different roles they play. Okay, very important topic. So when we say that these are the 5 classes of the immunoglobulins, you can remember it by a mnemonic MAD GE. Okay, so we start with the IgM, immunoglobulin M. That's how we call it. Ig means immunoglobulin. Okay. So what is an IgM? IgM is a pentamer, as you can see here. It's a pentamer, right? 1, 2, 3, 4, 5 antibodies are together here. Okay. And as it acts as a pentamer, right, what happens? It also acts as a monomer as well, but usually found as a pentamer. Okay. So what happens, as a monomer, it's used as an antigen receptor. What does that mean? That means if this is a B cell, okay, on every B cell you will have a receptor which can bind to a specific antigen, right? So this is a receptor. So this receptor is also an antibody, okay, and this antibody belongs to IgM class. Okay, that's a very important thing, right? So other than that, it also helps in the complement fixation. If you remember, we spoke about complement fixation last time, right? Even that is in your exams also now, right? right? How complement proteins, which are present in the plasma, how they are activated, right? And if you remember, we spoke about how they form, they assemble complements, they assemble with each other, and they form a channel within the plasma membrane. And through this channel, the contents of the cell will come out, or the bacterial cell will come out. And that's how the bacterial cell is bursted, right? We call them as a membrane attack complexes. We spoke about that, right? So yeah, that's called the complement fixation. And this IgM is very much important in the complement fixation. So the question says that name a immunoglobulin which is a pentamer and which can fix complement. So you can say it's the IgM. Is it clear to everyone about IgM? Okay. And then we have the IgA. Okay, IgA acts as a dimer. Let me show you. Look at this. So it's something like, let me get in the color. As you can see here, there's one antibody here, two antibodies lined up together. Okay, it's called the dimer. Okay, and this is your IgA antibody. This is again a very important antibody because whenever we talk about sweat, intestinal juice, milk, saliva, all these things are rich in IgA antibody. Very important, right? So they're very limited in the plasma. They're usually found in these body secretions, okay? And they're very specialized. They stop the pathogen, from attaching to our epithelial cells. If I inhale air like, okay, if there are, some foreign toxins present in the air and they try to go into my epithelial tissue, okay, in my throat, right? So IgA antibody is going to stop that, okay? It will not allow them to attached to the epithelial tissue. So that is the specialization of the IgA. So the question says that name the antibody which is found in the body fluids, in the intestinal juice, milk, etc. So you will name it as the IgA antibody. Then comes the IgD. Okay, a very simple. It acts as a monomer. Monomer like this only. That is how it acts, like monomer. Okay, and it is found on the B cell. Again, it acts as a receptor on the B cell. If this is our B cell, okay, so the antibody which you find on the B cell, which can recognize the antigen, that could be either IgD or either IgM. Okay, so this antibody does not have any other role to play, so it's very, very important only in the activation of the B cell, right? Because B cell will be activated once it will bind to the antigen on its receptor like this. Then this B cell will be activated, will form a plasma cell, and then they will form the antibodies of this type, right? This type. So same antibodies will be formed by the plasma cell. Is it clear till now, guys? Everyone? What is IgD? Okay, then comes to a very, very special kind of antibody, which is called as the IgG. It's very, very special. Many questions can be asked about it. Okay, so what is an IgG? Why is it special? It's special because it's the most abundant, almost 75 to 85% of the antibodies, circulating in our blood plasma belong to IgG. Like, so very abundant, right? So this is a very important antibody because when it comes to primary response and the secondary response, we spoke about it last time. When a person is exposed to the antigen for the first time, like coronavirus for the first time, it's the primary response. And then if he's reinfected again by the coronavirus, that is the secondary response. So in both the responses, in both the cases, when a person produces antibodies, it belongs to the IgG antibody. Okay, and this antibody can also fix the complement like your IgM, as we discussed here. And the most important thing about IgG is, excuse me, this is the only antibody which can cross the placental barrier, right, and reach to the baby. I would like to share something interesting with you. Did you hear a recent news that a female who was pregnant, okay, and during her pregnancy, she was given a vaccine, okay, against the coronavirus? Okay, did you hear about this news recently? It was not here in Saudi, it was in some part of the world, right? She was given a vaccine for coronavirus. And when the baby was born after 9 months, the baby was born with the coronavirus antibodies. Did you hear about this news? No. So how did the antibodies reach to the baby? So that means the baby which was born with the antibodies, okay, those antibodies were of this class, IgG. That means the baby had these IgG antibodies because these are the only antibodies which can cross the placenta and reach to the baby. Exactly, exactly. So can you believe usually when the babies are born, they are born with what? With the innate immunity, right? So as they grow up, as they're exposed, they drink milk and are exposed to the air, right? They are exposed to more and more antigens, and slowly they developed the humoral immunity. They're born with the innate immunity. But then when the antibodies are given from the when the antibodies pass from mother to baby, okay, this is basically the passive immunity which we spoke about last time. Remember, the natural passive immunity from mother to the fetus. This is an excellent example of the passive immunity. So look at this, the baby is born with the passive immunity, but the antibodies will stay in his blood for some time, right? And soon he will be, or he, she, I don't know whether it was a she or a he, okay, but I read it somewhere, right? So you can relate whatever you study nowadays, okay, you can relate it with your real life, right? So now you know that the antibodies, exactly, right? So see, I feel like giving all these updated information to you so that you can relate your whatever you're learning with the life, what we are living right now. Okay, so there were many people who said that how is it possible, how can baby born with the antibodies? So if you are an anatomy student, if you are a doctor, you know that IgG antibodies can cross the placenta, barrier. These are the only antibodies which can cross the placenta, right? And thus giving the passive immunity from mother to the baby. Then comes the last type of antibody called an IgG antibody, right? This is very, very important for the allergies. Okay. If, for example, I inhaled a bacteria or I inhaled dust particles, I will quickly start sneezing, right? Or the watery fluid will come out of the nose, right? So that's kind of an allergy which happened instantly, right? So this is the antibody which is formed at the time of allergies. So their level rises so very much at the time of the allergies, okay, whether the allergy is in the skin, whether the allergy is in the GI tract, whether the foreign body is in the respiratory system, right? So that is the kind of antibody which you find it during the allergies. Okay, then we discussed all this. Then we just will, okay, no need to discuss, no need to memorize the table because the same information is already in this slide. So no need to memorize the table. Okay, this is the last slide which we're going to discuss about, and then we can move on to the revision. So antibody functions. Okay, so again, I'm discussing here so that you don't forget it forever that antibodies cannot kill the microorganisms, right? They can simply inactivate them. They can simply tag the antigens. For example, this is an antibody which will bind to the antigen. Now, this antigen is inactivated by this antibody, right? It forms an antigen antibody complex, antigen antibody complex. Now, this antigen is in trouble because it has been inactivated by the antibody, and now antibody cannot do anything. Antibody cannot kill this microorganism, but then what will it do? It will become, these complexes will become easier targets for the phagocyte. And the phagocyte will come from here. It will start engulfing this hole, right? But the antibody simply inactivated it. It simply marked it for, destruction, that you're going to die now. Okay. But then it's the phagocyte, or maybe the T cell, or maybe the complements, which can actually kill that particular microorganism. We'll first talk about the complement fixation. We spoke about complement fixation last time also, when we said that about the opsonization. If you remember last time, we spoke about that if this is a bacterial cell, okay, this bacterial cell is opsonized. That means complement proteins which are present in our plasma, okay, they coat on this plasma membrane, okay, and because of these complement proteins, the red ones which you see here, it becomes sticky. The bacterial cell will become sticky, right? And it will become an easier target for the macrophage, can easily eat this. Okay, that's what we learned in the complement fixation, right? So same thing here again. If you see here, the antibody has inactivated the bacterial cell by opsonizing it. Look at this. This is the bacterial cell which has been, inactivated by the opsonin. So here the opsonin is not the complement. Here the opsonin is the antibody. Now the macrophage can easily eat this complex, right? It can easily bind to it and eat away this. But then we also learned about this one, right? So once you have an antigen and the antibody complex, right, it is identified by the complement proteins, which are the plasma proteins, right? So as they identify this antigen antibody complex, they start becoming activated. As they become activated, they assemble around the plasma membrane of the bacterial cell and form a channel over here, as you can see here, right? And once the channel is formed, many channels will be formed on the plasma membrane, right? Everywhere the channels will be formed. So when the channels will be formed, the internal fluid will come out and the bacterial cell will be bursted, right? So this was the membrane attack complex. This is called as complement fixation. Is it clear? Wherein the complements were activated just because we had an antigen and the antibody complexes. Any question about it? Is it clear to everyone? Any question about the complement fixation? So you can say the complements are activated only when antigen will bind to that's like one of the pathways. They can be still activated if there's no antigen antibody complex. But here we said that when antigen and antibody will bind to each other, that will activate the complements. Okay. Then we have the neutralization. What is a very important these reactions? What is a neutralization? Neutralization is that, the reaction of an antibody. Okay, the reaction of an antibody. This is an antibody with the antigen. Okay, blocks, or you can say neutralizes some of the bacterial toxins. Okay, let's say for example this is a coronavirus. We all know that coronavirus has spike proteins, right? We all know that now. Even my 5-year-old son can identify now, oh, this is a coronavirus, right? Because we all know that they have spike proteins, right? So what happens as they have spike proteins, for example, what does a neutralization reaction mean? That the antibodies are going to come, they're going to block these toxic things, because they will use these spike proteins to bind to the target cell. For example, this is my cell, right? So coronavirus will use these, for example, binding sites. Okay, it will use it to target my cell. So before it can bind, the antibody is going to block all its toxic things, right? So as these toxins are blocked, so do you think now the coronavirus will attach to our body cells? Is it in a position to attach to our body cells now? Yes, ladies? Yeshatha, Gada, Noor. No, it's not in a position. It's not in a position to attach, isn't it? Because all the toxic areas of the coronavirus have been blocked by the blocked by the antibodies, and this reaction is called as a neutralization. Then comes your agglutination. Agglutination is basically, which you might have seen, whenever for example, if I have a blood group A, okay, and if I receive blood from the person who has a blood group B, isn't it? So my body, yes, Gada, please, dear, go. No problem. So what is the agglutination? Agglutination is basically, for example, if I have a blood group A, that means on my RBCs, I have A antigens, right? I have only A antigens. But God forbid if I've given with the blood group B, okay, which has B antigens on its plasma membrane, right? So this will be considered as a foreign, right? So my body will not take this, it will be considered as a foreign, okay? So immediately my antibodies will start agglutinating these all RBCs. RBCs will be agglutinated, okay? What is an agglutination? Because we know that antibodies have two or more sites for binding. For example, these are called as sites for binding, right? These sites can bind to the antigens. So they don't have only two, they can have more than two sites for binding, okay, to the antigen, okay. So what will happen? These antigen antibody reactions will cross link the pathogen to one another. As you can see here, this is an antibody, this is an antibody, this might be my RBC which I got from the from the other person when I was given blood group B, right? So maybe this was RBC with the B antigen, B antigen, B antigen, right? So this there's all B antigen on the plasma membrane, but my blood group is A. So quickly my antibodies will identify that, oh, this does not belong to us. Look at this, they have bound to their antigens and they have cross-linked them. Okay, they've clumped them, they're clumped to each other, okay, which results in the formation of these clumps, and these clumps will be easily identified by the what macrophage, and macrophage is going to eat away all these complexes of the RBCs along with the antibodies. Okay, this is called as the agglutination. Did you see in the blood, when you did the blood testing in the blood experiment, did you see when he added, for example, in the first drop of blood, okay, when he added the anti-sera A, or did he show you the clumping of the RBCs? It looked like broken RBCs. Did you see that? Yes. Unfortunately, we could not do this experiment because of coronavirus in university. But students do see that. Oh my God, these are clumped RBCs. You see that in the lab. How they are clumped by the antibodies. So the reaction is called as the agglutination. Then we have the precipitation. Precipitation is similar to agglutination. The only difference here is that antibodies are cross-linking the soluble antigens, not the whole RBCs as you saw here. Here, as you saw, the whole RBCs were clumped. But here, for example, if this is a blood and if it has soluble antigens here, okay, if it has soluble antigens here, so antibodies will come and they will start clumping them together. They'll start clumping them together, right? As you can see here, they've clumped them together. They've inactivated them. Same as, agglutination. But here you have to remember the antigen here is not the RBC, here it is the soluble antigens, okay, which they're too large to stay in the blood, and they're easily identified by the antibodies and they're precipitated out. Okay. Have you ever seen a precipitate in the chemistry lab? Have you done any experiment in the chemistry lab where you were adding something and it got precipitated out, it standed out of the solution? Exactly. It's the same thing, right? For example, when these in the precipitation reaction, when antibodies bind to the antigens, right? So this will come out as a precipitate, but then this will be attacked by the macrophage and eat this whole precipitate away, thus killing the microorganisms. These are the diagrams for this, and that is all for today from the chapter lymphatic system. Do you have any question about today's lecture? And then we can start the revision. We'll do the cell mediated immunity in our next class, okay, and which is on Tuesday. And by this weekend, hopefully, we'll finish the chapter, inshallah. Then we can start with the respiratory system. All clear? Okay, alhamdulillah. Should we start revising now? You said, that's good. No, I sent you an email, Gada. I sent you an email that day, if you remember. Are revising, so we are revising chapter number 12, okay? So people, even if you want to leave the classroom, that's perfectly okay. I'm just revising, okay? So, but if you would stay, then it will help you to, to revise the lymphatic system once again, isn't it? So, in lymphatic system, we spoke first of all the functions. What is the main function of lymphatic system? We said that the main function of lymphatic system is that whatever fluid is lost, if you remember, I explained you that when there's high blood pressure, okay, on the arterial end, because of the high blood pressure, the fluid escapes out of the capillaries into the interstitial spaces. Along with the fluid, the oxygen goes out, and what I know, the nutrients come here, like glucose and everything, right? But what happens? We want this fluid to be reclaimed back, to come back to the blood vessels, right? And then we spoke about that, about the osmotic pressure, which stays same throughout. Okay, so because of osmotic difference between the blood capillaries and the interstitial spaces, so because of osmosis, the fluid is reclaimed back at the end of the capillaries, right? But then we learned that what happens is that, so all the fluid does not go inside, it's not moved into the blood capillaries. Some fluid might be left behind in the interstitial spaces. And in order to bring that fluid back, we have something called as the lymphatic capillaries. Okay, if you remember, this was the diagram. So lymphatic capillaries, which can bring the fluid back into the blood vessels. And we also learned that the fluid is brought back where? If you remember, can anyone tell me? In the subclavian vein. This was the point, right? Through the thoracic duct, right? And the other one is the right lymphatic duct, right? So the right lymphatic duct will bring from the face, right side of the face, from the right side of the arm, right side of the body, and bring it into the subclavian vein. That's what we learned. Then we learned, lymphatic vessels are just like the blood vessels, but of course they have different differences. We have a differences between the lymphatic vessels, right, as well as the blood vessels. And what are the various types of lymphatic vessels? We said that the various types of lymphatic vessels are your lymphatic capillaries, your lymphatic collecting vessels, right? And then we have the lymphatic trunk, lymphatic duct. I think we had a slide for that, right? So anyways, so all these are, look at this, yeah. So we have the lymphatic capillaries, we have the lymphatic collecting vessels, then we have the trunk, and then we have the duct. Duct is like the largest of all, okay, which finally pours the lymph into the blood, right? That's what we learned there. And now what is the difference between lymphatic capillary and the blood capillaries, right? If you remember, what is your blood capillary made up of? Does anyone remember? The blood capillary, what is it made up of? Remember the tunica media, tunica intima, tunica externa. Do we have all the three tunics in the capillaries? We have only tunica intima, absolutely right. So what was the difference between the the arteries and the veins? A quick revision so that you know what is the difference between arteries and the veins. Arteries had, if you remember, we spoke about arteries. You have to remember it, point wise. Number one, arteries are thick walled, right? Thicker. Two extra elastic layers. Very good. So thicker, more elastic. So point number one is thicker, right? Point number two is that they're more elastic. Point number three was that do they have valves? Yes. No, we're talking about we did not reach to the I started with the scratch. We're trying to discuss the difference between arteries and the veins. No valves, right? So thick walled, very elastic, no valve. What about lumen? What about the lumen? Remember, the lumen was smaller in arteries, whereas in veins, the lumen was larger. Absolutely. Isn't it? So these are something, these points which you will remember that. So arteries are thick walled, very elastic, no valves, less lumen, right? And they are at a very, very high pressure. Don't forget that high pressure. So 5 points for the arteries at very, very high pressure. And they have extra elastic layers. Okay. But then we discuss the veins. Oxygen, yes, absolutely. Oxygen, because they're very close to the to the blood, to the heart, right? So they receive, the aorta receives the blood which is at very high pressure from the left ventricle. That's why they are more stretchy. That's why their tunica media is very, very thick. Okay, and that is what the arteries. And then we learned that in the capillaries, we just have one simple squamous epithelia with the basement membrane. So we just have tunica intima in the capillaries, right? And then in the veins, we spoke about that. Obviously, the opposite of arteries. So they are not thick walled. Tunica and tunica media is not thick walled. So less smooth muscles, isn't it? Veins have less smooth muscles. Number two, they have thinner walls because of this less smooth muscles, less elastic. They have valves at low pressure, isn't it? They're all at low pressure, right? And then we learned that veins have lowest pressure in the body, whereas the two superior and inferior vena cava has the lowest pressure. That's what we learned about the veins and the arteries and the capillaries. Now let's try to compare the blood capillaries with the capillaries of the lymphatic system. So what was the difference? We said that if the question says what is the difference between lymphatic capillaries and blood capillaries? First of all, again the same thing. They're just made up of tunica and intima. That's one thing. But what was the basic difference between lymphatic capillaries and the blood capillaries, guys? Tell me the different points. Closed at one end. Absolutely. Lymphatic capillaries are closed at one end. Your blood capillaries are not closed at one end. Very good. Point number two, more permeable. Absolutely. More permeable. That is why when the cancer cells can move into the lymphatic capillaries. Very good. Are they, are they pumped by something? Do they have any pulse? No pulse. No pumping action. No pulse. Okay. What about pressure? High pressure or low pressure? Low pressure again, isn't it? And not fed by arteries. They're not fed by arteries, right? If you see here they are fed by arteries. Look at this. The blood vessels are fed by arteries, but here they are not fed by arteries at all. Look at, they start as a blind ended vessels, right? So that was the difference between lymphatic capillaries and the blood capillaries. Now if you have to compare your lymphatic vessels, be it a duct, trunk, or vessels, generally we say that the lymphatic vessels, okay, they resemble to the veins, right? We just saw that veins have thinner walls as compared to arteries. So that is the same thing here. Lymphatic vessels will have thinner walls like the veins. Okay. Okay, one more thing we forgot about lymphatic capillaries. Nobody said about the valves. Guys, nobody said about the valve. Do we have valves in lymphatic capillaries? Yes or no? Yes, nobody said this point. There are no valves in the blood capillaries, but we do have valves in the lymphatic capillaries, right? And not only capillaries, we have valves in the lymphatic vessels as well, right? So veins also have valves. Lymphatic capillaries and vessels, they also have valves. Lymphatic vessels are at low pressure. Veins are also at low pressure. Veins have less smooth muscles or thinner walls. Lymphatic vessels have also less smooth muscles, thinner walls. Isn't it? So veins also have, they basically, for example, they have a very minimum pressure. So lymphatic vessels also have minimum pressure. Rather, they have no pulse, they're pumpless system. Okay. So that was about the differences between the blood vessels. Is it clear till now? Are we okay? So yes, so we said that the main function of the lymphatic vessel is that it has to absorb the lost fluid from the body. If that fluid is not absorbed into the lymphatic vessel, what will happen? There will be a swelling. Lot of fluid will be accumulated in the interstitial spaces. There will be a swelling, and we call that swelling as the edema. It will result in the edema, right? So that was about the function of the lymphatic vessel. That's what we studied first. Okay, and let's move on to the lymphatic nodes. Okay, the very important point, lymph node. What is the function of these lymph nodes? Anybody? Why do we have lymph nodes? What is their main function? Why do we have them? They have filtering what? Do they filter blood? Do they filter blood, Rawa? No. Yes, very, very important point. They do not filter it. Blood has nothing to do with the lymph nodes. Nothing to do. Filter the blood, Gaida. No, I just said that. Don't make this mistake. They don't filter the blood. They filter only lymph there. Okay, dear? They do not, they have nothing to do with the blood. They simply filter the lymph, and then that filtered lymph is brought back to the, look at this. So the lymph which was here, the lymph which was in the interstitial spaces, right? The interstitial fluid, it was taken in by these lymphatic capillaries, and finally, look at this. It passes through all these lymph nodes. On the way, it gets filtered, passes again, it gets filtered, okay? And then finally, the same lymph is brought through the thoracic duct into the subclavian vein. And then this filtered lymph will become part of the blood, or it will become plasma, right? So lymphatic node is, as I said, that it's made up of what? Connective tissue. So you will have connective tissue all around here, right? With various compartments in it. Okay, let me show you the diagram. And then we said, look at this. So this, let me show you. These are the various compartments called as the trabeculae present within the lymphatic nodes. Okay, and then we said that the outer cortex of the lymph node is very, very rich in these purple colored things, which are called as the follicles, which are nothing but the collection of T cells and the B cells. Okay, and then in the center you have this yellow colored thing, which is nothing but which is a germinal follicle, which is basically very, very rich in the antibodies. Okay, and then we learned that this is the medullary area. This is the medullary area, okay, present here. Okay, and then in short, we learned that in the medullary cord area, we also have T cells and the B cells. The cord is like, this one, this purple colored things which you see here. These cords are also rich in the T cells and the B cells. And medullary area is very rich in the phagocytic cells, right? So then we learned that the most important thing to remember here is they contain all the macrophages, they contain all the phagocytes, they contain all the lymphocytes, T cells and B cells, and its function is to filter the lymph. And then we discussed that they have more efferent, most important thing, they have more efferent vessels than the efferent vessels. The reason for that is so that the lymph which travels into the lymph nodes, it can stop here for some time, and it can be easily purified or cleaned or filtered. Then we learned about the other lymphoid organs like the spleen, thymus, and all. What was the main function of spleen, guys? What is the function of spleen? Why do we have spleen? If you remember, I told you it has a white pulp and it has a red pulp, right? Then what did we learn about the spleen? It produces, it basically, it's the most important point where the lymphocytes are proliferated. They encounter the antigens there, right? And they destroys the old age RBCs. Okay. They also form the blood cells in the fetus. Okay. And the spleen also stores the platelets. Okay. And some amount of blood in it. Okay. And there are certain macrophages which are present in the spleen, which can basically kill the microorganisms, okay, which are present in the blood. So right, it filters the blood at the same time. Is it clear to everyone? Then we discussed about the thymus gland. About the thymus gland, we simply said that the primary lymphoid organ, okay, which, can you name the primary lymphoid organs, guys? What are the primary lymphoid organs and secondary lymphoid organs? Can anyone name them? The primary are thymus and bone marrow, absolutely. And the rest, all of them are secondary. Remember, thymus and bone marrow are primary. Whatever you see after that, whether we talk about spleen, whether we talk about tonsils, whether we talk about Peyer's patches, lymph node, they're all spleen, they're all secondary lymphoid organs. And the largest, remember that, the largest of all is the spleen. Out of all the secondary lymphoid organs, the spleen is the largest of all, right? This is a very important point to remember. Okay. Okay, then we spoke about, so the T cells and the B cells are formed in the bone marrow, but for maturation they move to, T cells move to thymus for maturation, right? Because there's a hormone called as thymosin. Okay, while as B cells stay there in the bone marrow. Primary is where the T cells and B cells are formed, you know, where they originate. The origin of T cells and B, they originate in the bone marrow. Okay, so that's one. And then they also mature. Where do they originate and where do they mature? So T cells mature in thymus and the B cells mature in the bone marrow. So the primary lymphoid organs are those organs where the lymphocytes are formed, point number one. And point number two, where the lymphocytes are also matured or they become immunocompetent there. Clear to everyone? Right? Spleen is not a primary lymphoid organ. I said spleen is the secondary lymphoid organ. It's the largest secondary lymphoid organ. No, I never said spleen is primary there. Only remember that bone marrow and thymus is the primary lymphoid organ. It's okay, no problem. Right? And then we have the tonsils, right? Tonsils, as we know that they are protecting our respiratory tract, right? These are the tonsils, right? And they simply remove the bacteria by trapping them, and then they are engulfed by the macrophages all over there, right? Then the Peyer's patches. Where do we find Peyer's patches, guys? Which area do we have Peyer's patches? What do they guard? What do they protect? They protect our intestines. Absolutely, very good. They protect our intestines. You see the diagram over here. Look at this. Peyer's patches are protecting our intestines, right? And so are your lymphoid follicles in the appendix, right? And what is MALT, guys? MALT. What is MALT? MALT. Which organs are protected by MALT? What is the full form of MALT, guys? We spoke about MALT as well. Mucosa associated lymphoid tissue. Remember? Yes. So where do you find MALT? You will find all those area wherever you have a threat of bacteria to come in, right? So tonsils also form. They're also considered as MALT, right? So tonsils are because they protect our respiratory system. Lymphoid follicles and Peyer's patches, they protect our small intestines, right? So together, all these are called as the malt because they protect our those areas which are very rich in microorganisms. Then we discussed about immunity. We said we have an innate immunity and the adaptive immunity. In the immunity, we said that we have the, if you remember the table which we spoke about, in the innate immunity, these are the all these things which give us innate immunity, the physical barrier, the first line of defense, where the skin and the mucous membrane play a very, very important role, right? Other than that, we also have the saliva or the tears or the stomach mucosa, the hydrochloric acid, right? Or the mucin present in the mucous membrane, right? Mucous fluid. So all these give us the immunity for. So you should remember all these. So as I said that, you should at least remember this table. That was the main idea of making this table. So you should remember what all comes in first line and what all comes in the second line of defense mechanism. Okay. So other than that, and then we have the second line of defense. Okay. So in the second line of defense, we spoke about phagocytic cells. Yeah, you all can leave. Yes, I'm about to finish. I just have to discuss second line. People who have a class, please leave. Thank you. Thank you. Okay. So I'm just about to finish the second line defense. Yes, people who have a class, please leave, ladies. I wish you all the very best for the exams. Yes, dear. Okay. So here we spoke about, in the second line, we spoke about the phagocytic cells, right? Wherein how macrophages engulf the microorganisms and eat them away. We also spoke about natural killer cells. Who will tell me how do natural killer cells kill the microorganisms? Yes, I'm waiting for, answer if anybody is connected. They make holes, absolutely. What do they produce? I know that, yes, you're right, they make holes, but what do they produce? They produce two important things. How do they call lysis? The granzyme, yeah, legends. So they produce granozymes and perforins. Remember, we spoke about granzymes and perforins, right? This one, perforin and the granozyme. If you remember this, right? And these are the only cells which can kill the cancer cells and the virus infected cells. Very, very important point to remember. So no phagocytosis for the natural killer cells, not at all. Okay. And then we also spoke about inflammation, right? So we discussed all the what are the four signs of the inflammation: redness, heat, pain, and swelling. And we also spoke about how kinins are produced, okay, and histamines are produced, how they dilate the blood vessels, how they make the blood vessels leaky, right? And at the same time, as the blood vessels are leaky, right, the neutrophils also escape from the blood vessels. And why do they escape? And what is that process called? As the neutrophils, if you see here in the diagram, neutrophils, they escape out and reach to the area which is infected. So their movement is called as a diapedesis, right? We discussed that. The movement of neutrophils from the blood vessels to the area which is infected, we call that diapedesis, right? And finally, what happened? All these things are done so that the healing can become faster and faster. So inflammation is also a very, very important sign for the second line of defense. If the question says that, yes, Reem, you can leave, no problem. Okay, I will, yeah, I will be, yeah, I will upload it sooner, don't worry, inshallah. The moment it will be, I will receive the lecture recording, I'm going to upload it right away. Okay, don't worry, dear. If you have a class, you can leave, no problem. Okay. So that was about what? That was about the inflammation. And then we spoke about the phagocytosis. Yeah, we spoke about that, how they engulf it. Okay. And then we spoke about also in the second line defense mechanism, then we spoke about two important things: the complement proteins and the interferons. Okay. In the complement proteins, what did we say? We said that complement proteins are the proteins which are present in our blood. Okay, they're present in our blood, but they're always inactivated. Okay, complement proteins are always inactivated, right? So as they are inactivated, what is going to happen? What activates them? How do complement proteins activate? Can anyone tell me? They're activated by, does anyone know how they are activated? What activates the complement proteins? Yes, ladies. What activates them? Nobody? So they're activated simply by when they encounter with the microorganisms. That activates the complement proteins, right? So how do these complement proteins kill? They kill by various mechanisms. They kill by destroying the bacteria. They destroy the bacteria. And as they destroy the bacteria, they form the membrane attack complexes. We just spoke about that, right? This one, they form the membrane attack complexes. That's how they destroy the bacteria. And second thing, the mechanism how these complements protein kill. The second thing was that they promote the phagocytosis. They promote the phagocytosis. How do they promote the phagocytosis? As if you remember, I told you that these complement proteins, right? We just discussed about that. As this is a bacterial cell, it will be covered by the opsonins. As it will be covered by the opsonins, and this bacteria will be easily engulfed by the macrophages, right? Because it becomes sticky now. So adhering to the macrophages become easier. So the whole thing will be engulfed by the macrophages, right? And the third thing in phagocytosis, and the third thing is that they also, during complement activation, they also cause vasodilation. Vasodilation. As they cause vasodilation, so a lot of neutrophils will escape out through diapedesis, right? And because of the chemotaxis, right? So all those things will be sent to the area of the infection. That is how the complement proteins have various ways of killing the microorganism, either by destroying them, either by activating the phagocytosis, or causing vasodilation or chemotaxis. And then for the interferons, we said the interferons are very sweet. We said that if this is the cell, okay, which is a healthy cell, this is a healthy cell, okay, this healthy cell will produce interferons I, okay. As these interferons will be produced, I, okay, then we have here, sorry, these are not healthy cells, these are infected cells. I'm sorry, these are infected cells, okay, infected cells, okay. And then if you have here healthy cells, these are healthy cells over here, these are healthy cells over here. So the infected cell, which is infected by the virus, that will produce the interferons, and these interferons will go and bind to the healthy cells. As it will bind to the healthy cells, now these healthy cells will be what? They will produce the antiviral proteins, and they will be ready for the attack if virus will try to attack these cells as well. So interferons will not protect the cell which is infected, but yes, interferons will protect the other healthy cells. That is what we learned till now in this chapter. With that, we revised the whole lymphatic system till now, right? So any questions? Any other question?